this is some of the most compelling new data that's come out in a long time. And I'm sure pharmaceutical companies are not too happy about it because it's difficult to patent and profit off of something like this.
Hello, everyone. It's time for another episode of the Ramos Law Difference Makers podcast, where, as your host, Dr. Jim Hoven, I get a chance to talk with cool people doing amazing things, making a difference, just like we want you to make a difference in every part of your world so that together we can change the world. And today, I am so excited for the episode that we're about to bring to you. In fact, this is such incredible information. Some people call it controversial. Some people call it groundbreaking. That we're going to make this a multi-part venture so that you will hear part of our topic today, and then we're going to move on to where you'll hear more about the topic in another episode, or maybe even two. So this is going to be really exciting. You're going to have questions about this. You're going to have thoughts. For some of you, it might get your hair standing up in like, whoa, what are we talking about? For others, it's going to get your hair standing up in anticipation. So as I've teased this topic out a little bit, let me tell you what it is. We're going to be talking about psychedelics, not as a specific, or sorry, not as a generalized category, but one in specific that our guest, Dr. Don Cooper, and you've heard him before. Dr. Cooper is a mad smart guy. And not mad as in angry, but mad as in ridiculously smart. And he is very versed in the topic of psilocybin, which is what we're going to talk about today. So I'll have Dr. Don once again. He's been with us several times. He'll give you a little bit on his background. We'll talk history. We'll talk effects. We'll talk theories. There's going to be a lot to this next series. So buckle up and hang on. You're going to want to share this information with your friends. And so let's get started right away. Dr. Don, welcome back. This must be like episode 5 or 6 we're doing together at this point.
No, it's fun.
And by the way, love your shirt, man.
The
Psycho Bunny,
yep.
coming at it. So for those of you watching this on YouTube, you'll notice Dr. Don looks dapper as always. And so Dr. Don, as we go into this, there's a lot that we can talk about, everything from the origins of psychedelics as a class of substance all the way to specifics. But in general, as we go into this, I think if we laid the groundwork to say that for eons, civilizations have found a way to use plant medicine and herbs and other things in order to help them cope with life, deal with pain, deal with stress, and sometimes even reach what we'd call spiritual enlightenment. And this class of compound, as a psychedelic, psilocybin, is part of that process that's been used by indigenous peoples and different tribes for a long time, right?
Right.
And as part of that, again, that's not necessarily the focus here, but why people go into this is completely individual. Mm-hmm. Some people recreationally, which mm-hmm. is probably not the best idea. Some people medicinally, some people even spiritually mm-hmm. is why they take compounds like psilocybin. And there's a lot to it. We want to unpack that today. Go through just a little bit about your background first as a neuroscientist and your history as you've come to find an interest and an expertise in this area.
Sure. Well, one thing I'd like to say about that is that if you really look at all of the drugs of abuse that are taken, if you look at the coca leaf, it comes from a plant. If you look at nicotine, it comes from tobacco, which is a plant. You look at psilocybin, it's a fungi. So all of these things really come from plants that the thinking is that they were initially defense mechanisms for plants against insects. And as we evolved, we still retain, if you look at the fruit fly, for example, there's a lot of common neurotransmitter signaling that goes on in a fruit fly and the same type of plasticity that we have in our brains. And so it's interesting that if you look across all these different plants, that they have such still powerful effects on us as mammals as they have on insects.
Isn't that something? Like, I always think about this, and I don't know if you've ever wondered this as a true scientist. Like, I'm not even a pseudoscientist. I'm just a guy whose brain floats around a lot. I thought, who's the first person to say, I think I'm going to try this plant? Out of nowhere, out of nothing, you take the plant and you take it, and all of a sudden you're like, wow, I'm feeling this, or I don't feel that. The history of that would be interesting to learn at some point. You know what I mean? Like, that's just so wild to me.
It's an interesting study for anthropologists.
Mm-hmm. And maybe we should go have one of those guys on and find out what's going on. But as we dive into this, I'm going to ask some basic questions. And I know that you are again, your versatility in this topic is going to be wide range. So I'll just start going through some processes that we've worked on and questions that we've talked about. But if this wants to go anywhere outside this, please feel free to take it, because I know our audience is really excited.
I think that, you asked me the question about my background, and how I came to understand or to be interested in these drugs. And it was really, I was here at the University of Colorado Boulder at the Institute for Behavioral Genetics with a guy by the name of Al Collins, and he was a prominent figure in understanding the genetics of nicotine and alcohol. And as an undergraduate, I worked in his lab, and I was always asking him about dopamine and the reward system. And he got so fed up with that he called up one of his colleagues at Chicago Medical School, name of Frank White, who studied the electrophysiological basis of drugs like cocaine and amphetamine and how they affected dopamine signaling and the brain reward system. And he says, look, I've got a guy to send to you as a graduate student. I think that you guys will mesh really well. So I went to Chicago Medical School and studied some of the first generation of genetic knockout mice, looking at D1 dopamine receptor signaling and its effects on and how cocaine goes through that signaling process, looking at single cell recordings. So this is really understanding how drugs like amphetamine and cocaine and even some psychedelic drugs, how they affect the dopamine reward system and the prefrontal cortex, and in general, cellular decision making. And then after that, I spent about 5 years getting the PhD doing that, published with a couple Nobel laureates on the first generation of knockout animals. And then I went to Northwestern and spent about 4 years there looking at, specifically with Nelson Spruston, looking at dendritic integration and how prefrontal cortical neurons and hippocampal neurons, how they process information and how they propagate information within a single cell. And then I'm also studying drugs like cannabinoids and cocaine and the effects of plasticity, repeated cocaine administered to animals, and then how it changed their excitability of these networks.
So basically how substance in leads to function out,
right,
through the nervous system.
Right, and how you could have, the main question that I had during my thesis was trying to understand how individuals, using an animal model, but how individuals who are repeatedly exposed to cocaine, in this we're using self-administration in rodents, where they can actually press a lever and get an intravenous injection of cocaine. We felt that was very important to understand the willful administration versus just an experimenter coming up and injecting them in the belly. And that had a different component, engaged a different network where the animal can pace it, and the animal can decide whether they want the cocaine and how much they want. So we were really interested in how this process of self-administration over days, where we would allow them 6 hours at a time to do nothing but take cocaine, how it would change the excitability and processing within certain areas of the brain, like the prefrontal cortex and the hippocampus. And we were focused on those areas because in humans, researchers, Nora Volkow, who's head of National Institute of Drug Abuse, she had shown that in humans that are cocaine addicts, that there is what's called a hypofrontality, where there's a reduction in the glucose utilization or processing energy utilization in the prefrontal cortex after repeated cocaine in cocaine addicts that have taken cocaine for a long period of time. And the consequences of that were that they had an inability to process contingencies, and by that I mean risk. So if someone says, well, you probably shouldn't do some behavior, you probably shouldn't speed because you might get pulled over, you probably shouldn't do cocaine because you might be arrested, that's a prefrontal cortical processing task, and it's part of this whole what's called top-down inhibition, where the prefrontal cortex kind of dampens the subcortical areas that are involved with drive and motivation. I want something, right? So if your prefrontal cortex is not functioning properly and it's hypofrontal, then you simply cannot factor in the risk side of that equation. And so all you experience is the drive. And that's essentially what happens with drug addicts. Is
of all types? Like with alcohol and
alcohol, there's hypofrontality. There's even loss of neurons, death of neurons. Methamphetamine, same thing. Cocaine, amphetamine. You don't get that with psychedelics. So we can talk about why that is, but specifically psychostimulants, alcohol, you don't get that with nicotine. Okay, so what you get with the drugs that really cause problems with decision making are drugs that affect the prefrontal cortex, and that's the area that really is the highest evolved structure in the human brain. It's what separates humans from other organisms, is the sophistication of that prefrontal cortex, and it's what enables us to decide what's worth pursuing, what goals to pursue, and also it allows us to interact socially. And that's really one of the primary things that separates humans from other primates and organisms, is our ability to network and integrate with other people and empathize with other people. And that's really a prefrontal cortical task. And if you have changes resulting from repeated exposure to cocaine or taking cocaine where you're addicted, you don't have that ability, then that causes a pretty substantial disruption across the person's entire life.
Right.
And so I was hoping to do is I was hoping to understand at the very cellular level, how is it that we can take these fMRI images, which is in humans, where you're looking at oxygen utilization in the prefrontal cortex, and the fact that it goes down, that tells you one thing, but what we still didn't understand why. How is that possible that repeated cocaine could do this and last for days and months and perhaps years after the person's taken cocaine? There obviously was some kind of plasticity that had gone on, and learning and memory.
And so when you are going through that, you're seeing this. Was there some point in time where you discovered, number one, did you already know that psychedelics as a class did not hammer the prefrontal cortex like these other drugs? Or what got you interested then in now turning the page onto psilocybin and what the other psychedelics?
Come back to psilocybin. Well, the fact is that at that time we didn't know anything about psychedelic drugs. They were much more taboo and very difficult to research. So I was focused on trying to understand how dopamine signaling and modulation could affect prefrontal cortical excitability, and try just to understand how neurons process information under the influence of, bathed in different types of dopamine signaling and serotonin signaling. So we had very rudimentary tools at that time, and what came out of that was really a better understanding of what cocaine and amphetamine do, as opposed to, and we can contrast this, because I think it's very important to understand the distinction between those two classes of drugs, in terms of if we talk about ego dissolution, which is something that happens with psychedelics. But if you think about what happens with psychostimulant drugs, they cause grandiosity, they cause increase in ego and increased confidence, and they cause an ability to filter out noise and increase the signal that's coming in. They help you focus, and that's why in some ways therapeutically, ADHD, attention deficit hyperactive disorder, is treated with low doses of amphetamines and other drugs like methylphenidate or Ritalin that preferentially increase dopamine in the prefrontal cortex. And what that allows them to do is tune out a lot of distractions and focus and engage an attentional network, right? But what you can think about when it comes to psychedelics is that they kind of do the opposite, in that they add noise to the network, they increase connectivity with lots of other brain regions, and they promote kind of this. They actually decrease signal to noise. And if you really think about the subjective effects between someone who's taking cocaine and someone who's taking psilocybin, those are radically different experiences, subjective experiences. But I mean, that's just we can get to all of that. It's just that my background went from trying to understand the cellular mechanisms of plasticity at the measuring the electrophysiological states of neurons, to then when I went to UT Southwestern as an assistant professor, I was in Eric Nestler's department, who had really founded molecular psychiatry. And he had studied, he built a department based on a kind of a genetic basis of transcription factors and how they participate in drug abuse and plasticity and mood disorders. And so there I started really getting fascinated with how genes and gene transcription factors could influence plasticity. And one of the main problems that hadn't been addressed in terms of plasticity and memory formation that I was interested in was, at that time, we knew that in the cellular models of memory formation, there was a phenomena called long-term potentiation, which was something that, it was a model that showed how neurons end up becoming strengthened and connected to where they can form a memory. But that there was a problem with that, in that it could only explain memories that were formed over minutes, hours, or days. The type of memory I was interested in was more the types of memories that are short-term and ultra-short-term memories, the types of memory traces that give continuity to our experience and our consciousness. And there would be the type of memory where, let's say we were engaged here in this conversation and someone were to come in the door and interrupt us and ask you a question, and then they would leave. I would still be able to remember exactly where I was in the conversation. I'd be able to resume exactly where we left off. But that's only tens of seconds. And that's not enough time for our neural networks to engage with increased firing activity, calcium cascades leading to transcription factors, leading to mRNA, then translation of proteins, shipping the proteins, and then restructuring and remodeling neuronal dendritic arbors. I mean, that takes minutes to hours. So how is it that we can possibly remember a name in a party, over tens of seconds, and just the things that give us continuity of our experience? There was no cellular model, or there was no real model to explain how those memories were even allowed. And so I became fascinated with the mechanism of these short-term memories that kind of give continuity to our consciousness. And out of that, I was focused on some particular type of channel called transient receptor potential channels, and they're specifically expressed in areas of the prefrontal cortex in these large layer 5 projection neurons, glutamatergic neurons. And these are really interesting in that they're activated by a number of different compounds, serotonin, psilocybin, for example. And once they're activated, they can stay firing and they can turn the neuron on and make it fire for up to a minute without decay. And so they can hold a memory trace. This is what allows neurons to maintain a memory trace for up to a minute without any network activity whatsoever. So in 2009, I published a paper on that in Nature Neuroscience, which was essentially a cellular model for of memory formation and how individual neurons in the absence of a network could maintain a memory trace. And I think the most analogous way of thinking about what this is like is, ask yourself, is it possible for you to know that you'd just eaten a hot pepper? How much processing is required for you to know that you just ate a hot chip, or that what do they have, the chip challenge, the one chip challenge?
Yes.
Where you have that. I haven't tried that myself, but I know in principle what it does, it activates capsaicin receptors, and those receptors are very much like the ones I described. They're transient receptor potential vanilloid. They're called TRPV receptors, and they're in the mouth. And what happens is they start these calcium cascades, and they turn on these neurons, and they just keep firing. And so even if you drink water and you rinse it out, you could still remember 30 seconds ago that you ate a chip, right? Mm-hmm. It doesn't require any higher level processing because that memory is coded in your tongue.
Yes.
And so it's that we have the same types of receptors that code for that type of capsaicin and these transient receptor potentials in the prefrontal cortex. And so since we have that, we have the ability for this type of sensory memory trace in individual neurons. And I think that's important for when we're studying things like hallucinogens, anything that activates serotonin, a particular receptor, the 5-HT, it's called 5-HT2A receptor, because it activates the same signaling cascades that you have with capsaicin in your tongue. And so it allows you to maintain these like memory traces in the absence of new information.
Wow.
So, that's kind of the background on how I can come into my interest in understanding how these types of drugs can be used, how they modulate different neuronal networks at the cellular level. So,
and what's interesting about this is we are talking. You use the correct term. We are talking about drugs. Some considered medication, some considered not medicinal. And so one thing that we're definitely going to let people know is obviously these are the hallucinogens, the psychedelics, psilocybin. They're not legal everywhere. In fact, they're not legal a lot of places. And so the topic of our conversation is not to go through and what should be done and what is done and where can you and where can't you, but rather just to inform and educate on the science behind how these compounds work and what they do to our bodies and what patients have experienced who report on them and that kind of thing. And so we certainly want people to that we're not condoning people do any treatment, especially in this area, without having some significant medical attention and supervision. But I think us giving the information that you're going to share is going to be really important. And I think that's a good place for us to start with. Now kind of springboarding from the background and everything you shared neurologically from how you got here and your interest to now talking about the mechanical side of it, of how does psilocybin work in our brain? What exactly does it do once ingested? Okay,
so the psilocybin is usually taken as a dried mushroom. So typically the amount of psilocybin in a magic mushroom would be around 1%. So if you have a 1 gram mushroom, then you have about 10 milligrams of psilocybin. And what happens is when it's ingested, it's actually what's called a prodrug. And that means that it's a drug that needs to be converted into another form, into a metabolite, before it can exert its effects. And so psilocybin is dephosphorylated by alkaline phosphatase, and it's also subject to breakdown by acids. So when it hits the stomach, it starts to break down into psilocin. And it's really psilocin that does all the heavy lifting. It's the one that crosses the blood-brain barrier and activates 5-HT2A receptors. And those are serotonin. There are about 7 different types of serotonin receptors. And they all have different signaling cascades, cellular signaling cascades, and they all have different distributions in the brain and body. And the ones that are predominantly located in the cortical areas of the visual cortex, auditory cortex, and prefrontal cortex are the 5-HT2A variety. And that's where psilocin is activating. Now, psilocybin, it takes about an hour for it to exert its effects, and that's because it has to be converted into the psilocin through the liver and also in the stomach.
And is there a psilocin direct, like is there something that someone could get from a doctor or something of psilocin, or all psilocin comes from converted psilocybin?
Well, it's kind of like heroin. Heroin is also a prodrug, and it's converted to morphine. So it's something that you can convert it to psilocin. You can take psilocin directly, and it'll have a faster onset. It's just that it's one more step that the body has to do in order to get it to that activated compound when it's ingested and it's in its natural form. So typically, though, there are probably if there are probably different formulations that one could make in order to facilitate that conversion, but in general, the main heavy lifting is done through psilocin.
And with this serotonin system, can you go into that a little bit as far as practically, what does that mean? We got these different receptors that you had spoken about, and then you've got the psilocin coming up and being absorbed by them or connected to them. What does that result in? Is that where the kind of hallucinogenic effects or any of that start coming in? Is it through that particular connection right there?
Yeah, the nice thing about, one of the things that is a worthy goal in pharmacology is getting a very specific drug, one that doesn't have side effects. And the nice thing about psilocin is that it very specifically, selectively binds to 5-HT2A receptors. And those receptors activate they're called G protein coupled receptors. And when they bind, they activate a signaling cascade intracellularly that activates calcium. It increases intracellular calcium levels, and that calcium is one of the most important signals that neurons use. It activates ion channels that cause electrical excitability of neurons. It turns on gene transcription factors that lead to mRNA and protein translation for reorganization of memory machinery. So it does a lot of, calcium is a really important signaling mechanism. And so these particular receptors are located in high density in the visual cortex. They're high density in the prefrontal cortex. And so what happens is when you can selectively activate those receptors, then you're going to get modulation of inputs that are particular to those areas. And if you really think about the serotonin system is involved with almost everything. It's involved with sleep. It's involved with appetite. So if you look at appetite suppressants, drugs like fenfluramine, fen-phen, they activate serotonin. It's also involved with mood. So Prozac, that's a drug that's an antidepressant. Selective serotonin uptake inhibitors are what's used to treat depression and anxiety. So serotonin is released from an area of the brain that's close to the brainstem called the raphe nuclei, and it's a series of different nuclei that send projections throughout the brain, and it goes to the areas of the hippocampus, the temporal lobes, the prefrontal cortex. It just goes out throughout the brain, and it's involved with almost, like I said, a variety of different systems, but just the receptors, the 5-HT2A receptors, are highly localized in specific areas, and that's what gives it the unique signature of a psilocin or psilocybin versus, say, Prozac.
Okay. So when we're looking at these receptors, you talked about a kind of a 10 milligram dose, based on the gram of the mushroom and this kind of thing. I know that I've also heard, and you and I have talked about a couple of information sources, podcasts that we've both listened to, and they talked about something called a hero dose, and the hero dose being more along, along the lines of a good bit more than that, and then I think it was a 10, a 25, and a 50 was the dosages that they were using. So you just referred to the 10 version, and that 25 was where they were finding a lot of really good effects. And the hero dose was kind of being done outside the scientific community. Right. And they were going through stuff. And so when we look at this, if we look at the common experiences of feelings that people report after taking this, does it depend on the dosage of a standard versus the little larger versus this hero dose versus even a micro dose? What is is that tied to what the receptors can take in? They get flooded, or how does all that work?
Right. There's a dose dependency of almost all drugs. So a microdose would be something that is designed to be beneath the person's subjective awareness. So they could take it and they may not be aware that they're taking it. And that would be on the order of maybe 1 milligram.
Mm-hmm.
And then 10 milligrams would be a threshold dose where you would start to see some effects, maybe you would experience some subjective effects. And then 25 is where they found the most therapeutic benefit in a single dose in intractable, depressed subjects. So people who have been on other types of antidepressants where they've failed, the research is showing that about 70% of people who have already been on something and failed can take it one or two times at 25 milligrams and go into remission.
That is impressive.
So it's one
70%.
of about 70%. And so this is some of the most compelling new data that's come out in a long time. And I'm sure pharmaceutical companies are not too happy about it because it's difficult to patent and profit off of something like this, But it is definitely something that is promising.
When you first started your research on this, you'd mentioned earlier that there was nothing known about this. Like there was just not a lot known. And I remember as kind of from some side material I was learning, and then with some of the information that you and I have talked about, you have people like Timothy Leary and Richard Alpert over at, I believe they're at Harvard, and they were psychologists that were going through their process, and then they started taking these things and they were reporting like, man, listen, I've learned more about the human mind and psyche in a matter of a couple of trips or journeys, as the state might go, than I did all through medical school and now working with these folks.
Yep.
But then it was seen as in a different context where it was dangerous, because then you had the, tune out, drop out kind of thing that was going on culturally in the hippie movement and all the rest of it. And so these compounds got tied politically to danger versus, and it got rid of our natural curiosity on how they work towards our benefit. We're in a different space now, right?
Yeah, I think so.
Yeah, where people are like Johns Hopkins and these places, there's big studies going on at prestigious universities on how to use this for things like depression and things like that.
There's even one at the University of Colorado in Anschutz.
Beautiful.
They're looking at this.
And so this gives us a sign of hope in the future. And I know we'll get more to that, but kind of now taking maybe half a step back from this dose dependent, and as we talked about it for medical conditions such as depression, if someone just takes these things, what are the things that they'll feel if they were to engage in a psilocybin ingestion, whether it be from obviously past a microdose, because they're not going to feel anything in a microdose, but from that 10 up past that milligrams, what's the body feel?
A lot of this was done by a guy by the name of Alexander Shulgin, where he published a couple books. He and his wife went out to the desert, and they synthesized a lot of different tryptamines and psychedelic compounds, and then they would take them, and then they would catalog their experience.
So when you say they synthesized them, they created them, or they got them from the plants?
He was a chemist.
Okay.
He would extract. They would synthesize their own. They would take them at different doses. They would describe the subjective effects,
like all good scientists do.
Yeah, they were the original psychonauts, explorers of that internal space.
Wow.
But I mean, if we were to quantify this, I think that now we know that in the therapeutic setting, there are a couple common things that happen to most people who take these at the specific doses in a therapeutic setting, say around 25 milligrams. And that is that usually the onset occurs within 45 minutes to an hour, somewhere in there. And what they see is sometimes there's an initial anxiety, so there's some kind of uneasiness. There may also be a little bit of nausea, and that's just simply because of that conversion of the psilocybin into psilocin. It turns out there's a lot of serotonin in the gut. Actually, the highest concentration of serotonin in the body is in the gut. It's a different source of serotonin than in the brain, which comes from the raphe nuclei, but still there are receptors there. And so it can cause some kind of nausea. But then that usually, sometimes they refer to that as hitting the wall, where there's this initial kind of unease and anxiety. And then there's this kind of it transfers over into, well, let me just say this, that in the therapeutic setting, this is almost always paired with music. So when you go through a therapeutic session, you start off with very soothing sort of music, expansive music, and then it goes into something that's meant to be very calming. Mm-hmm. And then as it moves, there's more of a feeling of emotion. And so, and a blending of different sensory inputs with sound and vision. You start to see patterns in the visual patterns. And then there's also what's called synesthesia, which is this sort of crossover between a blending of different senses, like sound and vision tend to be linked in some way, or experienced as one thing. So that'd be like the
plant starts singing kind of thing? Is that what that would express itself as?
It's kind of like you could rub your hands. You could rub your hand and manipulate the experience, the tone of the music. It would go. It would just, the pitch might change.
Wow.
Or when music is playing, you're looking at a light and the intensity of the light would sync with the music.
Okay.
So that's called synesthesia, where two senses seem to be working in concert as one. And so then there's that for a while. There's can often be, this is usually guided with two health practitioners that are licensed psychologists, where the person might be talking about the emotions that are coming up. A lot of times they're childhood memories or deep-seated emotions that they have that they can express and talk about and explore. Or it may be something where they're just commenting on the information that's coming in, the sensory information, and so they can go back and forth. And it's kind of interesting to see this, because you can see the individual. I talked a little bit earlier about this top-down inhibition, where there is this role of the prefrontal cortex to control the amygdala, an area that's involved with emotional expressiveness and the sort of fight or flight stress response and anxiety, and it's subcortical. It's very primitive, and the prefrontal cortex can put that into context, and that can help the person. But when you have someone where you have this struggle of the prefrontal cortex, they tend to, the ability of the cortex to regulate that is somewhat dissolved. And so they can go back and forth between experiencing euphoria and happiness to sadness within seconds.
And I remember from looking at some other information on this, you talked about music being a very important part, and that the music changes to where in the height of the experience, which can be, I don't know, a couple hours in or something, that there's a lot of drum and kind of real beat-based stuff. And then as you come down off of your journey or your trip, then it goes back to this kind of softer music based on just where you are neurologically and emotionally. Is that right?
That's right. It matches the time course and the pharmacokinetics of the peak levels and what people tend to experience and where it hits, what structures it hits, what systems it hits. And so you do have this music that goes into bringing in more drums, that's more activating, it's more energizing. So there is a component where they feel energized. And then there's this element of what's known as ego dissolution, where there's kind of an ego loss. And what that simply refers to is and this can be stressful for some people. And that is where they start to lose their ability to distinguish themselves from their environment. And so the music, they experience this as being one with the lighting, one with the music, one with other people, everything seems to be blending together. And so there's a loss of, if you will, signal. And if you contrast that, as I said earlier, with individuals who are taking recreational levels of cocaine, that's entirely grandiosity and goal driven and focus and high ego. It's almost the exact opposite. And if you play the same type of music for someone who's taking cocaine, they would find it probably very annoying, because there's just all this information in there, and they would listen to maybe one aspect of it, and they would focus in on just one specific component. But the neural processing for what goes on with a psychostimulant is fundamentally different than what goes on with a psychedelic.
What are the other clinical uses that we're finding or that we're studying that these compounds could benefit folks from?
Cancer, people who are end of life. There seems to be a dramatic effect for that